All right, welcome to today's webinar by SCIEX, and I'm introducing here today the new SCIEX 4500 series LC-MS/MS systems and their applications in clinical research and forensic toxicology. So here we have the new SCIEX triple quadruple 4500 and Q-Trap 4500 LC-MS/MS systems, which are the instrument of choice for clinical research and forensic toxicology laboratories that require a dependable, integrated, workhorse LC-MS/MS solution to deliver fast, sensitive results that they can trust hour after hour and day after day. Now, this new system includes many of the features that make its big brother, the 5500 series mass spectrometer, so popular. And these include an integrated valve, an integrated syringe pump, the legendary Turbo V ion source, and a very small footprint, which is only 32 inches by 32 inches in width and length and 24 inches high. This system also supports up to 180 pounds of UHPLC equipment on top of the mass spectrometer. So both this feature and the small footprint make the system ideal for laboratories where space constraints are a concern. Now, the 4500 system has big shoes to fill. It builds on the legacy of the API 4000 system, which was arguably the most successful triple quad ever. This new system builds on the legacy of the API 4000 by setting a new benchmark for reliable quantitation with at least 10 times greater sensitivity over all other triple quadruples in the same class. The published specification for 200 femtograms per serpene on column is a signal-to-noise of 2,000 to 1. Now, there are many new innovations that result in improved performance in the 4500 series mass spectrometer. The first of these is the QJet2 ion guide. Now, the QJet2 ion guide is an RF-only quadrupole which effectively focuses ions into the Q0 region of the mass spectrometer. And what this does is it captures more of the ions than a traditional skimmer cone would capture. Allowing more ions and less gas into the Q0 results in much higher sensitivity. In keeping with the tradition of the API 4000 system, the 4500 series of mass spectrometers has an excellent robustness, and this is in large part due to the Turbo V ion source. This robust source features an orthogonal spray design and supports flow rates of up to 5 mils per minute. It also allows easy-to-use plug-and-play electrospray ionization and atmospheric pressure chemical ionization probes. In the slide presented here, we can see robustness test for the compounds methamphetamine and amphetamine. 1,500 injections of plasma were made over 7 days, and we can see that the internal standard corrected peak areas of these compounds were less than 3% and less than 2% respectively for amphetamine and methamphetamine. Another key feature of the new 4500 series LC-MS/MS systems is the new vacuum system with only one small turbo pump. In comparison, the 4000 series had 2 turbo pumps. The new interface design of the 4500 series with QJet2 ion guide enables much more efficient pumping. This new vacuum system is not only more robust and reliable for more productivity, but is a much cooler and quieter system. Another improvement in the new 4500 system is the EQ electronics. The upgraded electronics allow improvements in both speed and extended mass range. In terms of speed improvements, this system supports 4 times faster tandem quadrupole scan speeds compared to the API 4000, with no loss in quality in precursor ion and neutral loss scanning modes. The system also supports fast MRM measurements with dwell times of as low as 1 millisecond and pause times as low as 1 millisecond. Furthermore, this fast system supports positive-negative ion polarity switching in 50 millisecond switching time. In terms of mass range improvements, the new RF coil box design with a drive frequency of 940 kilohertz provides a mass range of up to 2,000 Daltons. The higher frequency better confines the ion beam and especially improves the sensitivity for lower mass ions. And finally, there have been improvements made in linear accelerator trap technology. These improvements include axial field within the linear ion trap, which move ions towards the extraction region. Also improved is the ion extraction itself, and much more efficient in-trap fragmentation. The faster scan speeds and cycle times of this instrument allow scan rates of up to 20,000 Daltons per second, with better than than unit resolution. There are also much shorter in-trap fragmentation times, all of these allowing this instrument to run well in compatibility with UHPLC methods. Another key feature of this instrument is the curved LINAC collision cell. This space-saving design allows for a smaller instrument footprint. The new linear accelerator design is what enables the short dwell and pause times and the fast scanning in MSMS mode. Furthermore, the curve of the LINAC collision cell helps to eliminate neutrals, preventing noise on the detector. This system also features new detector technology for quantitation. The accurate pulse counting detection system counts pulses of ions impinging on the electron multiplier for a timed period, creating digital representation of ion flux. This system is less susceptible to noise than analog detectors. Furthermore, it does not require filtering and thresholding of data. All of this means that we get better detection of low-level signals with the highest possible accuracy. Now let's look at a few examples of the performance of the new SCIEX triple quad 4500 system. In this first example, we'll investigate the quantitation of 6 compounds along with 6 internal standards in a single method using the MRM mode. Comparing the sensitivity of the new 4500 system to the 4000 system, we can see that on a compound-dependent basis, we get improvements in sensitivity that range from 1 to approximately 3 times in intensity. If we scale the data for the 4000 Q-trap system to the same axis, we can see that the improvements on the Q-trap 4500 and triple quad 4500 system are quite significant. On this slide, we have a summary of a direct comparison of the sensitivity in MRM mode, comparing the triple quad 4500 system to the API 4000 system. As you can see, the gain in signal for these 5 compounds range from 1.1 times to 2.4 times, with an average gain in signal of 1.6 times. All in all, the SCIEX triple quad 4500 system delivers equivalent or better sensitivity compared to the API 4000 system. On a compound-dependent basis, nevertheless, we expect that, by and large, we will see performance gains. And just to demonstrate the analytical and quantitative performance of this new system, this slide demonstrates the linearity, accuracy, and CVs for quantitation in MRM mode using one of the example compounds that we were investigating in the previous slide. For the antiretroviral compound, M-tricidabine, over the range from 5 to 5,000 nanograms per mil, we can see that the accuracy is never more or less than 10% deviant from the expected accuracy, and the CVs range from 1 up to 7%. In this second example, we investigate the use of the Q-trap 4500 system for the purposes of drug screening. In this example, we've taken urine and spiked it with a mixture of drugs. We monitor 508 MRM transitions simultaneously, 2 transitions for each of the compounds under investigation. Because of the turbo-V ion source, we're able to run the system at a flow rate of 2.2 mils per minute. In this particular experiment, we will leverage the speed of the Q-trap system to perform fast, sensitive scanning, allowing collection of high-quality MSMS spectra for every compound detected. In this slide, we can look at some of the data collected with the method described previously for the analysis of drugs in urine using the Q-trap 4500 system. This analysis was performed using a Phenomics Kinetics C18 column and a simple dilute and shoot sample preparation. We can see that while screening for 508 transitions simultaneously, we have detected the presence of a number of compounds here. And we have simultaneously generated EPI, or enhanced product ion spectra, for these compounds. We have representative spectra for hydromorphone, meperidine, and fentanyl. Not only does the Q-trap 4500 system give us incredible gains in speed, it also provides incredible gains in sensitivity when operating in linear ion trap mode. On the left-hand side, we have comparable data acquired using a 4000 Q-trap system with the MRM survey scan on the top and the IDA-triggered MSMS data on the bottom. When we compare to the same experiment performed on the Q-trap 4500 system, we can see that we have a moderate gain in sensitivity in MRM mode, as expected, but we have an enormous gain in sensitivity in EPI, or enhanced product ion mode. Much more signal, much cleaner spectra, which means much better library searching confirmation. And in this third example, we investigate the possibility of performing positive-negative switching with scheduled MRM. In this example, urine has been spiked with a number of drugs, including pain panel drugs, barbiturates, and the metabolite of THC. We know that the pain panel drugs will fly in positive ion mode, while the barbiturates and the THC metabolite will fly in negative mode. The speed of the 4500 system allows us to monitor both the positive and the negative compounds simultaneously using the scheduled MRM algorithm. On this next slide, we can see an example of the quantitative performance of this system while running a positive-negative switching experiment performed using the scheduled MRM algorithm. We use buprenorphine as a representative compound, and we can see that the excellent linearity over a range of 0.25 to 1,000 nanograms per mil is more than acceptable. On the right-hand side, we've tabulated the percentage CVs and the accuracy over the entire range. We can see that the accuracies range from 84% to 117% over this range, and the percentage CVs are no worse than 12% at the low end of the concentration range. The 4500 series LCM-SMS systems deliver more than just a tandem mass spectrometer. These are just one part of a total solution. The SCIEX Accelerated Lab Integration Package for Clinical Research combines the 4500 series with the Exogen Expert UHPLC systems, the Cliquid software, iMethod applications, multiplexing capabilities with online solid phase extraction, and training into a comprehensive workflow specific to high-throughput targeted quantitation. So in conclusion, the 4500 system delivers the world's most popular triple quadrupole intelligently re-engineered. This fit-for-purpose design offers best-in-class performance with equivalent or better sensitivity in MRM mode compared to the API 4000 system. This system offers a mass range up to 2,000 Daltons. Many speed improvements have made the system compatible with the fastest chromatography, and these include the fast dwell and pause times of 1 millisecond, the 50 millisecond polarity switching times, fast quadrupole scan speeds up to 12,000 Daltons per second, even faster Q-trap scan speeds up to 20,000 Daltons per second. The Q-trap has a 50 times improvement in sensitivity in Q-trap scanning mode compared to the existing 4000 Q-trap. Furthermore, the speed and sensitivity improvements on this platform lead to vastly improved quantitation in MRM cubed mode. And finally, we have several improvements such as the integrated syringe pumps and valves and smaller footprints that lead to improved usability of the system. Thank you very much for your attention.